Okay, everyone, I have 12 o'clock, so we are going to get started. Thank you so much for joining us for our 2025 Virtual Water Ambassador Program. The Water Ambassador Program is a partnership program between Martin County, University of Florida IFAS, and the Florida Sea Grant Program. This is our lineup for our upcoming presentations. We will be putting the registration link for each of our upcoming topics in the chat. So in March, we have a presentation by Martin County, Michael Houston, talking about seagrass restoration efforts in the county. Then we're going to be having a sort of a multi-speaker collaborative presentation on oyster aquaculture, approach to clean water and local economies. And then in May, we are going to have another sort of collaborative approach where we're going to be talking about Martin County land acquisition and watershed restoration with a presentation followed by an expert panel for you all to have some question and answer time with the folks who are doing land acquisition for watershed restoration in the county. We also have a field trip coming up on Monday, this Monday, to the South Florida Water Management District C44 Reservoir and Stormwater Treatment Area. This is part of the larger Comprehensive Everglades Restoration Project, and this will be guided by the South Florida Water Management District. The registration link can be accessed either through this Google form or at the QR code. I will tell you that we have a limited number of spots left. We are almost full to capacity. So if you want to join us, register now because we will be closing registration very soon. As always, this webinar will be recorded and hosted on the Martin County IFAS Extension YouTube page. So this presentation, as well as all of our other previous webinars, are found on that YouTube page. We'll be putting that link in the chat as well. And we will be holding all of our questions to the end of the presentation. So feel free to type them in the chat. And once the presentation content is done, we will then go through the question and answer with our speakers, which I am really excited to share with you all today. We have multiple speakers today, all coming from Dr. Carrie Reinhardt Adams' lab at the University of Florida, and they are going to collectively be talking about the right plant for the right job, Spartina alterniflora, and Florida's living shorelines. So we've got a suite of bios to go through, so I'll run through them real quickly before I turn it over. So Dr. Carrie Reinhardt Adams is an associate professor of plant and restoration ecology with the Environmental Horticulture Department at UF. She studies the transition from invasive species dominance to native species establishment and focuses on approaches to scale up experimental manipulations to become applications in natural resource management at the landscape scale. Her research addresses questions in plant ecology with conservation and restoration applications, with case studies in wetland, salt marsh, and submerged aquatic vegetation plant communities. Madeline Estes is from Jacksonville, Florida, currently pursuing her master's degree in interdisciplinary ecology with the School of Natural Resources at UF. And her research includes germination and greenhouse experiments, as well as surveys of nursery growers and restoration practitioners, which will identify challenges and potential solutions to the production, sale, and acquisition of Spartina for living shorelines. Stasia, I'm gonna leave Stasia to pronounce her last name on her own time because I'm not even going to attempt that. Stasia is from Sarasota, Florida. She graduated from Florida State University and is now at UF with mentored by Dr. Carrie Reinhardt Adams and Dr. Laura Reynolds, working in plant and restoration ecology with a focus on seed dynamics of Spartina in Florida, understanding how we can better utilize direct seedling methods for restoration. And last but not least, Tori Guarino is a first-year PhD student. At UF, Tori works with the Restoration and Plant Ecology Laboratory, focusing on living shorelines. Tori plans to assess genotypes from natural systems of Spartina to understand how sourcing from natural environments may provide the traits advantageous in living shorelines exhibited by Spartina for the native plant industry. So, you are all fortunate enough to hear from all of them today because they're going to be doing the joint presentation all about Spartina and living shorelines. So with that, I'm going to stop sharing my screen and turn it over to Carrie and her graduate students.
Thank you, Lisa. Yep, it's a 4-for-1 today. Let me get the right slides up here. Okay, and good on the slides. Can you see my first slide?
Yep, looks good.
Awesome. Okay, great. Well, thanks so much, Vincent and Lisa, for hosting us today. We're going to talk about the right plant for the right job, Spartina alterniflora in Florida's living shorelines. And there's just so much to study about this plant. I'm excited for my students to tell you about their work. They're actually going to talk to you today instead of like me talking and taking credit for their hard work, which is really not fair. So yeah, we're at University of Florida. I'm Carrie Reinhardt Adams. You'll hear from Madeline, Tori, and Stasha, who are in my lab. I also want to acknowledge our UF collaborators, Laura Reynolds from Soil, Water, and Ecosystem Science, also Christine Rohal, who is part of that department, as well as my department, which is Environmental Horticulture. And then Xiao Yu from Engineering, who has been helping us look at how different traits of Spartina affect sediment trapping in coastal environments. I also want to acknowledge that none of this stuff could happen without our Florida Sea Grant Extension agent partners, Holly and Carolyn and Vincent, who have been great to help us with this project, providing their perspective and just making sure that our research stays really practical and useful. Also want to acknowledge our funders before we get started. The Indian River Lagoon, NEP, has been great to fund our research, Sea Grant as well. And we even have funding from the Florida Nursery Growers and Landscape Association because there's a lot of interest in actually growing Spartina for these projects. And also I wanted to note that this group is probably very well-versed in the importance of living shorelines and all that they can do for the coastline. But as a review, we've got a couple functions that we are looking for from Spartina in these living shorelines. Basically, we're designing living shorelines to stabilize sediment, trap nutrients, and provide coastal resilience as well as habitat. And as water ambassadors, I'm anticipating that a lot of you have some practical experience and connections with living shorelines, and really like to hear about your experience after we get done presenting, because that kind of context is so useful for us as scientists. But yeah, so living shorelines come in lots of shapes and sizes, and scientists have done a lot of studying how the plants contribute to these functions that we desire from living shorelines. And sometimes they have mangroves, like in A here in the slide. Sometimes they have an oyster shell breakwater. Sometimes they have a granite rock breakwater. But Spartina, or Spartina alterniflora, or Sporobolus alterniflorus, use the updated botanical term, is often a part of these, no matter what it really looks like, that living shoreline. We're just going to call it Spartina for the rest of this presentation. So it's a major component of living shorelines. So a lot of people find it surprising that despite that, it can be hard to find the Spartina you need for living shoreline projects once you set about doing that as part of your coastal management. And we'd like to know how we can improve plant material availability. And just to point this out, this isn't just a Spartina and living shorelines problem. It's a national problem and even a global problem that we just don't have the plant materials that we need to do the revegetation that we want to. And so Spartina is a great case study for looking at this larger global problem. Madeline Estes is going to talk to us more about her research on improving plant material availability. I also wanted to point out that we know from our work and from other labs that Florida population traits differ with source. You can see in this family picture of some Spartina to the right, we had a source that has skinny leaves, two that have fat leaves, one that's exceedingly tall. And these different traits are linked to different functions that are provided by the living shoreline. So it'd be good to know what's the story on these differences. Do they have a genetic basis, or is it adaptation? Regardless of the source, it definitely suggests that diversity might be a good thing for plant sources. So it might be important to make sure that you've got the right source for the job, but also, given all of the many conditions that might be unanticipated as part of living shorelines, it might be good to use many sources that give you many different, a diversity of sources which give you many different advantages. Also, because what we're often trying to do is mimic what we see in nature, it would be great to know how Spartina actually spreads in nature. It was once thought that it was mostly clonal vegetation, that clonal revegetation, spreading by rhizomes, that's the only way it spread. But since then, genetic studies have shown that, seed probably plays a role, but we don't really understand exactly how that works. So Stasha's going to tell us more about that. Speaking of that, it's time to talk about seeds from Madeline.
All right. Thank you so much, Carrie. Again, my name is Madeline Estes. I am a master's graduate student with Dr. Adams' Plant Ecology Restoration Lab. And yeah, I'm going to talk about how we can improve and supplement plant availability within Florida native plant nurseries and with restoration practitioners by utilizing seed as a plant material option for Spartina alterniflora. So the current status is that most restorations source their Spartina from commercial nurseries or donor marshes, or also they frequently will take transplants from another marsh on their property and move them to the restoration site. But regardless of any of those options, usually they are in the form of mature plants that have been clonally propagated via rhizomes. So they might be in bare roots, plugs, or in 1 gallon pots, but they have been basically split apart from one or a few sources. And to talk about there's some advantages and disadvantages to using the clones. Clones are a pretty easy methodology. Despite being pretty labor intensive, which is one of the cons. Imagine going out to a marsh and digging up with shovels whole mature plants. Um, After that initial input of labor, it's pretty easy to produce by just breaking apart that plant you collected and putting them in pots with more soil. And it's also pretty easy to guarantee a certain number of plants with lead time. So if the availability of sourcing those plants, either from a nursery stock or from a donor marsh, if that is available to you, it's pretty easy to say, well, by this time, I can have this many plants produced. They also have the advantage of being pretty rapid in establishment at the site. So there's pretty solid pros for using clones. But the cons are that as the name suggests, a clone is going to be genetically identical to whatever the source it came from. So imagine that there's a couple of collections, either from the same site or maybe from two different sites. They get propagated a whole bunch of times and all get planted at another restoration site. All of those plants at that restoration site are genetically identical to each other, and that might be happening across the state. So there's a concern there for a decrease in overall genetic diversity. Again, that labor intensiveness that comes along with using clones, and plant availability can be limited. A nursery might not have enough plants to supply all of the requests that are coming in for Spartina alterniflora because living shorelines are getting to be so popular. So what our lab is interested in is the role that seeds can come into play, and address some of these cons. So seeds, you get a lot more sources that can be available for collection. Collecting seeds is a lot less damaging to the donor marsh, for an example, than digging up whole plants. So that can open up more sources available to you. Seeds are generally less expensive, whether a nursery is looking to purchase a bunch of seeds to increase their stock or a restoration practitioner wants to buy seeds to plant at their site. They're always going to be cheaper than whole plants in like a 1-gallon pot, for instance. You also get the potential to produce many plants from a single collection effort. So again, the example of like going out and digging up whole plants from a donor marsh, you're gonna be kind of limited by time and maybe by storage space. And there's a lot of effort there that goes into getting that number of plants. For that same amount of effort, you could get a lot more plants by collecting from as many seed panicles or flowers as you can as possible, because Spartina really does produce a lot of seeds. And seeds also benefit from pretty much, they're always going to be more genetically diverse because they're produced by sexual reproduction than a clone is going to be. But some of those cons is what my research is really focused on. We want to get them closer to matching those pros that come along with clones to make them a viable option for people. So we want to address those kind of low germination rates that we've seen previously in the literature, and that a lot of nursery growers have this perception that Spartina is really hard to germinate. So I have some germination experiments to try to look at that. We also know from the literature that Spartina requires a cold stratification period. So that's a time in cold storage to break seed dormancy. That stratification period kind of just adds another layer of complexity to germinating these seeds. So we wanted to look at that for Florida. Also, given that Florida is at the southernmost range of Spartina, we were curious of whether Florida Spartina required that stratification period like the plants in New England might. And there's also a lot less information available about propagating from seed with Spartina. So we wanted to create relatively easy methodology like there is for clones that's going to be widely available to people. We wanted to make seed production more predictable by doing those germination and stratification experiments, and hopefully produce plants that are high quality that we can expect to establish well in a field restoration. So for my experiments, I had to go out and collect a bunch of seeds. That was the first step. Actually, the first step before that was kind of understanding when the seeds were ready to collect, which was a bit of a learning curve. We identified all these different stages that the panicles or the flower goes through before they're ready to collect. And that all happens over the course of a month or so. So I went out, observed the plants, collected when they were ready. We brought them back to the lab, and we realized that it was going to be really important that we sorted for fill. Spartina makes a lot of seeds, and due to a variety of factors, they only some of those seeds are actually filled with an embryo. So you get a lot of non-viable seeds, and we wanted to sort those out. We used an X-ray and a light table. So the seeds were soaked in water, so they were somewhat transparent, placed on a light table that shone light through the seed. And seeds that were filled, like the one on the left, so the right picture, the seed on the left, is a filled seed. And we were also able to see filled seeds in the X-ray. So the picture on the left, those bright white seeds, those are the ones filled. And then we could tell that pretty clearly different from the empty seeds or seeds that had been damaged by mold or herbivory. So we only wanted to use filled seeds for our experiment. The seeds went through a stratification period, so we put them in a refrigerator for either 0, 1, or 3 months. And then they went into an incubator. So the box, the picture on the left, is a germination box. I put a bunch of seeds in there, put them in the incubator, and let them germinate in what would be like ideal conditions. And seeds that germinated there, we transplanted into a greenhouse experiment to study their survival and their growth over time, and how those traits like survival and growth, either shoot growth or leaf growth or below ground biomass, how those might differ between different populations that I collected the seeds from. And what we found was that seed fill differed between our different populations, but generally they were all kind of below 25%, which showed that seed fill is going to be a really limiting factor that might either limit a lot or maybe limit a little bit depending on what source you collect from. Germination, across the board, seeds benefited from a cold stratification period. So seeds that were stored at 3 months germinated faster, and more of the seeds germinated than if they were not cold stratified. So Florida seeds definitely benefit from a cold stratification period. And then the seedling traits, so those that germinated that made it to the greenhouse, they also all differed with source. So they had different rates of survival. Some of them produced more leaves or got taller or made below ground biomass. And the fact that all of these experiments that I conducted showed clear differentiations with collection source suggests that there's a genetic diversity might be really important for restorations, because you can kind of, hit a lot of these different strengths that the different sources have. It also suggests the possibility of an optimal population to collect from. So if you're trying to choose the right plant for the right job, there might be an optimal population that, if we can predict that, either with genetics or environmental factors, which we're going to be looking at and talking about more today, might help inform that. So our lab is also currently looking at additional methods to improve germination. We're looking at seed cleaning as an option to add to that seed-based plant production guideline that we want to produce. We're also looking at what causes the differences between sources, either are they environmental or genetic, and Tori is going to talk about that next. And these experiments brought up questions about what the roles that seeds play in their natural environment, and what role they can play in restorations, and how that might relate to potential direct seeding technologies. And Stasha is going to talk about that as well. So I'm going to hand it over with that. Thank you all very much.
Hi, I'm Tori. Thanks, Madeline and Carrie, for the introduction on living shorelines and Spartina's role. So I will be going over the regional differences between Spartina along both coasts of Florida. So to start off, my objective is to overall understand the differences between natural populations on both coasts of Florida and how we can use these natural populations to better cater towards living shorelines and their roles to mimic more natural marshes. So recently, within the month of February, I did a lot of field work. So this involved going to 5 of my 6 sites. So to start off, on the Gulf Coast, I went to Big Lagoon State Park, Cedar Key Preserve State Park, Warner Boy State Park, and then on the Atlantic Coast, we have Pumpkin Hill Preserve State Park, Tomoka State Park, and then the Indian River Lagoon Preserve State Park too. So all of these sites, they have coastal differences, they have latitudinal gradient differences, and then inherent site characteristic differences like salinity, elevation, and just there's a lot of differences between these sites, and my goal is to understand these differences and how we can cater towards living shoreline technology. So to do this, a field survey was conducted, and some of the traits measured were shoot height, leaf width, leaf length, and then leaves per shoot to understand the inherent characteristics from each site. So starting off with leaf width, we have the most northern Atlantic site and the most southern Gulf site being the winners in that sense, which I thought was very interesting, the regional differences and how they're both similar in that sense. And then with shoot height, we have similar winners, with those sites being the largest. And then leaves per shoot, we have Cedar Key being the winner in that sense, and that could be related maybe to disturbance patterns with the recent hurricanes. And then with leaf length, we also have Pumpkin Hill and Warner Boyce being the winners. So it's very interesting that each site has different advantages, and then how we can use these advantages to cater more towards living shoreline and the ecological services we want them to offer for our coasts. So with each of these sites having these differences, it's interesting to see what these differences are attributed to. So this could be genetics. Do I admit someone if they're in the waiting room? Okay, this could be genetics, this could be the age of the natural site, maybe regional characteristics that we discussed before, maybe disturbance history, and then just other factors that are unique to each of these sites. Okay, so To assess this more, I transplanted 10 plants from each of the sources to see if these differences observed in the field persist in the greenhouse under controlled experiments to see what's really causing these differences between the sites by eliminating all the variables. So for me, my next steps is a genetic survey. So genetically characterizing these sites to see if these differences persist, and then what the differences are between each of the sites, if there's more diversity within the sites, and if that's leading to more of the trait expression, and then how this is playing out in the field and in the greenhouse. Okay, thank you.
Hi, everyone. My name is Stasha. I'm the last one to be presenting. I am going to be talking about my research, which was looking at Spartina alterniflora seed dynamics and what that might mean for restoration. So a little background on seed dynamics, it's how seeds interact with their environment. So that can be environmental variables that I listed below, like vegetation type, the cover of vegetation, the sediment type, wind that impacts dispersal, nutrients of the sediment, any herbivory that's going on, and inundation of tides. All of those environmental variables could impact seeds' role in the Spartina ecosystem. And so you have your original population, which is represented on the left side of the chart that I made. And then you'll have seed availability that comes from that population. And then within seed availability, you have seeds that arrive and that are sitting on the sediment surface. And then either those seeds will get washed away or eaten or die. And then there will be seeds that are retained in the soil that happens through accretion. And then those seeds that are retained can germinate, potentially become seedlings, potentially become mature adults that can add to the population. And I specifically wanted to focus on environmental variables of vegetation type and vegetation density, and how those might impact seed availability, and also germination. So my objectives were I wanted to examine Spartina marshes and look for seed arrival. So specifically, I was looking at seed availability, and I was specifically looking at seed arrival and retention in different patches of stem densities, because you can kind of see in this picture, there's areas that there isn't a lot of vegetation growing for whatever reason, and it'll be in like the middle of the marsh. And in this presentation, I'll discuss my results for seed arrival because I have not done seed retention yet. And then I also want to investigate the impact of seed availability and stem density on germination and seedling traits in a greenhouse experiment, in order to understand the extent that facilitation and competition plays with seeds and other plants in the area. So going into looking at seed availability, I had sticky seed traps that I used to collect the seeds in sites. And I had two different types. I had aerial traps and ground traps. And then I gave you this little representation of what it looked like at my site. I had them spread out, and I used a transect in order to have different variation in terms of distribution along the marsh. And then I had traps placed in vegetated and unvegetated areas. And I kept these traps out there for the seed rain season, which is about 2 weeks. And so this is kind of more of what my traps looked like at the beginning versus at the end. So I had my traps from vegetated and cleared patches. And then in the middle, I have some photos of what my data looked like that I collected. So as you can see, I highlighted a few of them. Some are easier to see in pictures, but there were seeds arriving either by wind, which was supposed to be represented in the aerial traps, or by tide or gravity falling onto the ground traps. And it definitely varied by site. So I looked at 2 different sites, and seed availability was different between sites and between seed traps, which was pretty interesting. And then I was really interested in, of course, knowing if there were seeds arriving in these vegetated areas and unvegetated areas, because if there aren't any seeds or any plants growing in those areas, I want to know what sort of environmental conditions could be impacting why those seeds aren't germinating. Which leads into my second objective, which is investigating those impacts on germination. And so I am doing it this month, but I am creating a controlled environment in a greenhouse where I will specifically be looking at the role of stem density of Spartina alterniflora and seed availability on germination and seedling traits. And facilitation, if you don't know what that is, it's essentially how one individual can help another individual grow. And for this, or flourish. And for this species, it would be intraspecific because it's the same species impacting the same species. And yeah, that's my experiments. And then my next steps, I am going to be looking at seed retention. So looking at the seed banks, and I'm interested to see what seed bank looks like in different sites, and if vegetation impacts seed bank. And then I'm also interested in understanding the genetics within this population. So kind of looking at Google Earth images to see how much the sites have grown and to do genetics within the population to understand what amount of new growth is from clones or from seeds. And then also something that would be interesting to do in the future is looking at the differences between natural marshes and living shorelines. For seed availability in order to understand more the role that they might play in these different types of marshes, because there's so many different environmental factors that could impact seeds. And thank you. We will be taking questions now.
Great. Thank you all so much for that fantastic group presentation. It was wonderful. We have so far one question in the chat, and Vincent, I believe it was yours, so I'm going to let you ask the question while I put up the poll for everyone.
Great. I actually put a couple questions in there. Great presentation, by the way. That was really cool.
Thank you.
I was just wondering if root characteristics are looked at as well as a potential genetic trait.
I'm so glad you brought that up, Vincent, because it's really, really, really hard to look at roots, right? Like washing them and drying them and weighing them that's the way that we do it, and it definitely does differ.
Okay.
And in fact, not only does the biomass differ, but also the ratio of fine roots to thick roots differs with populations. So, yeah, we were like excited and disappointed to realize that, yes, we do need to keep looking at the below ground attributes of these plants. Because obviously, you know, what you're probably cluing into is, you know, they play a really important role in sediment stabilization.
Yeah.
And so you'd think that as these different root characteristics differ with the populations, it could confer different aspects of sediment stabilization. That's thank you for asking about that.
Yeah, no, that's really important. Well, the other thing, the reason I asked is we had a fantastic root biologist at our research center, our IFAS research center, in Fort Pierce. Unfortunately, he moved on. He got another position somewhere else, but maybe that's someone to still keep in touch with.
Oh, I'll have to get that name from you.
Okay. Lorenzo Rossi.
Lorenzo Rossi.
He's
Okay.
Italian.
I think he's now at Texas. So
Is
I
it
did,
Texas? Yep.
yeah, I did launch the evaluation poll. So participants, if you can take 10 seconds out of your time to complete that. And Vincent, can you confirm that it actually launched for me?
Yes, I see it.
Okay, perfect.
Yeah.
And Jason has a question. He asks, how is temperature stratification defined and how do you accomplish it? Is it just putting the seeds in a fridge for a month?
Yeah, okay, I can answer that. So we stored the seeds at 4 degrees Celsius, which I should have off the top of my head what that is in Fahrenheit. But 4 degrees Celsius is what we stored them at. And that was we have a big like walk-in cooler in our lab that is at a constant 4 degrees Celsius. But your like refrigerator should not be all that different. A refrigerator should also be able to stratify the seeds. You don't want them to freeze, so you don't want to store them in the freezer. Okay, it's 39 degrees Fahrenheit is what that is. But yeah, you don't want those seeds to freeze. And the other thing is that you do have to store the seeds wet. So if they dry out, the seeds will like desiccate and die. So they have to be stored wet at 4 degrees Celsius or 39 degrees Fahrenheit. We store them in salt water just to prevent mold growth, because you can get mold that grows in the water as well. But that's what our stratification looked like.
Thank you. Appreciate it.
Okay, I think our evaluation is working, and we have another question. Are there known interactions between Spartina and other plant species that are either good or bad for the Spartina?
Boy, that's a great question. And one of the things that I think about with Spartina and interacting with other species, sorry, Tori, I'm standing in front of you, is like Juncus rhamarianus, right? Black needle rush. It definitely abuts Juncus in many salt marshes. And so I'm kind of hoarding some plants to do some experiments about that. But I'm waiting to see what happens with Stasha's plants to see what kind of interspecific facilitation we might see between Spartina plants and seeds. And then I'm going to use her experiment as a model to look at that. But as far as, you know, we know that Spartina and other salt marsh species are highly adapted for that like small elevational gradient of that has a combination of elevation, a combination of flooding and salinity stress. And so plants tend to be generally, you know, along their specific gradient in salt marshes, but with rapidly changing coastal conditions, I think those interactions are going to become more important to trying to make predictions about how plants might be moving along the coastline, you know, which is going to have tremendous implications for sediment stability. So I bet that's where that question is coming from. And it's validating to hear, because that is on my list of experiments.
Very cool. There's some similar work, I think, you know, looking at biodiversity, interspecific biodiversity, that I think some people from Harbor Branch, FAU Harbor Branch did. I'll try to see if I can track that down for you guys.
Thanks, Vincent.
Okay, our next question from Stacy is, what is the best sediment type for Spartina? We had some fail along a rather mucky shore and was wondering if sediment was the issue or something else.
Another great question. I'm so glad we added that factor in an experiment. Yeah, you know, I was really surprised, but our results do match that observation. We planted 6 different sources in high organic matter sediment and sandy organic matter sediment, and all 6 sources they accumulated more above and below ground biomass in the sandy sediment than they did in the organic matter. That being said, some sources had a greater sand advantage than other sources. So that kind of backs up our story about how, you know, different plants have different advantages. And so it's important to pay attention to those sources, the source of that differentiation.
Great. Keith wants to know, have you explored the differences or similarities in fauna through sampling and cataloging throughout the geographic locations adjacent to the plants? And how does that relate to Spartina's success?
Wow, that is another really good question. This is a good group. I know that Laura Reynolds' lab has identified, I think working with Savannah Berry, they came up with some ways to look at the infauna that establish as part of newly establishing Spartina shorelines. And they have an excellent EDIS publication that describes how you can actually measure your own infauna. But as far as studies done on our region, I'm not familiar with any. However, I know in other regions, as you move up the coast, people have done studies. Actually, yeah, thinking about one in Georgia that shows that the diversity in the genetic diversity in the Spartina was related to establishment of infauna and the density of the infauna. So it's really important.
Okay, great. And Edith and Vincent and I will do a quick search for see if we can pull up that Edith document.
Yeah, I think it's called like how to monitor the function of your living shoreline.
Okay, we'll see if we can find it and we'll put it in the chat for you.
Yeah.
Bill wants to know, is there a preferred water salinity that's best for Spartina?
Preferred water salinity. Hi, Bill. What's nice about Spartina is that it tolerates a wide range of salinities, right? Am I saying this right? And so it's often something else that constrains the distribution of Spartina and not salinity. I mean, it's done in our experiments, whether you put it in 0, 10, 15, 32 ppt, there's very minimal differences in growth, but it does not compete as well at some salinities with other plants. So it's like you can't really design a salinity that's going to promote Spartina the most, but you can think about the plant assemblage that's around Spartina and whether or not it's going to have an advantage in certain salinities. So yeah, it's more about the other plants around it.
Okay, and I think I found the EDIS publication, Living Shoreline Monitoring, How Do I Evaluate the Environmental Benefits of My Living Shoreline? So that is now in the chat for you all.
Thank
And
you.
Casey has a question. Do you have longer term outplant data, so more than 1 year? And how does it differ between asexual and sexual seedlings?
Wow, that's good. Yeah, that's really the question, isn't it? Like if we're putting forth all this effort and energy to make sure that we're getting the right initial plant composition, how long does that actually last in nature? And I am not really sure anybody has. Is somebody writing this down for a good next thesis?
I was gonna say, sounds like
We will capture the chat for you, Carrie.
Thank you. But I know that, like, for instance, in prairie communities, they've shown that the initial species composition changes pretty quickly. And you'd imagine that in a dynamic environment like living shorelines, that it wouldn't be surprising if certain genetic compositions were maybe rose to the top and had a higher survival and establishment rating than other types of genetic signatures. It's true.
Fantastic. Okay, I think that's the end of our questions. We'll give everyone another minute if anyone's typing right now. But in the meantime, I want to thank you all again for a fantastic lab presentation. I really like this format. So thanks for thinking of it. And otherwise, hopefully we will see some of you on Monday for our field trip. The registration link is in the chat, and otherwise we will see you in March. Oh, can you please add the full citation? Jason, is that for the EDIS? Or what's the full
Yeah, that's
citation
right.
for what?
I was on the link and it didn't go through for some reason. Do you have to be signed in on the UF server to get that one?
Nope, it's a public.
Yeah, it seems my browser's not making it to there all the way for some reason.
I will put the title in for you, and then you should just be able to
Perfect.
throw that into Google.
Okay, thanks.
Okay. All right. Well, thank you, everyone. And thank you, Carrie and Sasha and everyone else.
Alan, Tori. Yeah, thank you, students. You could, I'm glad you like this format, but you can really only pull it off if you have good, hardworking students who do a great job presenting their slides.
And you all did. Appreciate it. All right.
Thanks,
Take care,
Lisa.
everyone.
And thanks, Vincent, for the invitation. We really appreciate it.
Yeah, no, thank you for accepting it. And
yeah, great getting some
great
feedback from this group too.
Yeah, very cool. So, and a reminder that we will post this webinar. It's being recorded and it will be posted on our YouTube channel. That's the UF IFAS Extension Martin County YouTube channel. So keep up on that, and we've got our first one up from January, and then also look back at all of our past webinars.